السلام عليكم. محاضرتنا اليوم بالMOD هي Neoplasia 3. Why and how do tumor occur? The objectives of this lecture is to understand the inherited susceptibility to the development of tumors with specific examples, and then the inheritance of certain tumors with also we took certain examples of malignancy like familial adenomatous polyposis, breast cancer, retinoblastoma. The third objective is to know about the function of certain types of genes and their role in the development of neoplasia, mainly oncogene, tumor suppressor genes, and genes involved in DNA repair mechanism. The fourth objective is to understand the role of specific types of oncogenes and their related cancers. The fifth one is to know about the stages of carcinogenesis, and then we will take a specific carcinogenic agent, for example, radiation, chemicals, viruses, and other factors. We will talk about occupation, some types of occupations and their role in the development of certain tumors, and then we will go to know about some medical condition that is associated with a higher chance for development of malignancy. Finally, we will say something about geographical distribution or geographical variation about the incidence of some malignant tumors. So to start with, why do tumor develop? The tumor develop or arrange because of two factors اللي هي intrinsic and extrinsic factors. Intrinsic factors mean something inside the body, while extrinsic factors refer to outside the body. It means the environment. Mainly, the tumor develop because of interaction of intrinsic and extrinsic factors, not just intrinsic, not just extrinsic, but mainly interplay between the body and the environment. So, what is intrinsic factors? Intrinsic factors include inheritance, اللي هي genetics, and also host factors, mainly age, the immune system of the body, and the hormonal level or hormonal state. So, what is inheritance? Why it is important in the development of cancer? Because most, if not all, of the discovered cancer has disturbance in more than one genes, mainly the gene involved in the oncogene, tumor suppressor genes, and DNA repair gene. So, if we have disturbance in these genes, the body will have an increased susceptibility to develop malignancy. And here is some examples. This table includes certain malignancy associated with defect in the genes involved in the DNA repair mechanism. For example, retinitis pigmentosa, ataxia telangiectasia, and Fanconi anemia. Another examples of genes altered in specific type of cancers, example, the polyposis coli associated with disturbance in the adenomatous APC, which is adenomatous polyposis coli gene, and this is the chromosomal location of these genes. Another cancer is hereditary non-polyposis coli, which is associated mostly with defect in the mismatch repair gene. Leigh-Fraumani syndrome with disturbance in the p53 gene, and this is very, very important gene, and we will talk about it later in this lecture. The other malignancy is familial breast cancer or ovarian cancer associated with the BRCA1 and BRCA2 genes disturbance, retinoblastoma with abnormality of the RB gene, and we will talk about it later. We have two types of retinoblastoma and the difference between them. Now, the extrinsic factor, as we said, the extrinsic factor is anything outside the body, I mean the environment. So it includes radiation, chemicals, viruses. So why radiation is important? Is there any evidence for the role of radiation in the development of cancer? The answer is yes, there is. For example, the radiologist, I mean people work mainly in the radiology, has an increased risk of skin cancer. What else? Unprotected miners معناتها عمال المناجم, they work without protection, so they are exposing to a higher level of ionizing radiation. Later in their life, they have a tenfold rise risk for development of bronchogenic carcinoma. Another evidence for the role of radiation in carcinogenesis is that a group of children who have head and neck cancer, they receive radiotherapy. They found that these children developed thyroid cancer as a result of radiation. The other well-known example, the Hiroshima bomb, people who are exposing to this bomb develop earlier develop hematological malignancy, including leukemia and lymphoma, and later they found that they develop what is called sold malignancy, example breast and thyroid cancers. So all these are evidences that radiation may increase risk for the development of different types of cancers. How radiation produce carcinogenesis? So, as we know, the radiation will affect the DNA. DNA composed of two strands, and each strand composed of different nucleotide. So, the damage that resulted from radiation either affect the base, just base, or single strand or double strand. And the effect of radiation depending on the dose and the quality. So lower dose, the therapeutic dose, result in genomic instability, while higher dose will result in different kind of abnormality, genetic abnormality, maybe a mutation, different type of mutation like translocation, deletion, point mutation, whatever. So it depends on the quality and the dose of radiation. Another carcinogenic agent is chemical. The chemical carcinogen interact with DNA so that they will produce DNA damage. They may cause specific base damage or single strand break damage. Rarely it will result in double strand, not like radiotherapy, because we said in the radiotherapy we will have either base damage, single strand damage, or double strand. In chemicals, mostly chemical carcinogens result in base damage or single strand damage. Rarely result in double strand damage, and this is very important. Chemical carcinogenesis involve two steps: initiation and promotion. So it is very important to start with initiation. It means that the normal cell, when exposed to chemical carcinogen, it will be changed into initiated cells as a result of the effect of the initiator. This change is rapid and irreversible, but it's not enough to be transformed into neoplastic cells. So the cell must pass into another step of chemical carcinogenesis, what is called the promotion. Now the promoter will have the effect on these initiated cells in order to be changed into neoplastic cells. However, promoters by itself not produce neoplastic changes. It needs to be preceded by initiator in order for the cell to be transformed from normal to neoplastic cells. So again, we have the normal cell. In order to be transformed into neoplastic cell, it must pass through the initiation, then the promotion. You have to notice that the sequence is very important. When the cell exposed to promoter and then initiator, this will not result in carcinogenesis. The chemicals are divided into two types according to their ability to produce cancer. So these types are directly acting chemicals and indirect acting chemicals. Directly mean that they doesn't need metabolic conversion, metabolic or enzymatic conversion. So direct, there will be direct action to produce cancer. These are called weak carcinogenesis. Example, cyclophosphamide and chlorambucil. While indirect acting agents, from the name, it means that it needs metabolic enzymatic conversion. So that they will become stronger in order to produce malignancy. So we have specific examples of chemicals that are indirect acting. What we mean by indirect acting? It means that it needs metabolic conversion. So they are strong acting chemicals. For example, the coal tar that are present in the cigarette smoke. So here, by itself is not enough to produce malignancy. There will be hydroxylation so that the inactive component will be converted into the active product in order to produce certain types of cancer like lung cancer, bladder, skin cancers, esophageal cancer, different types of cancer associated with smoking. Another example of chemicals, indirect acting chemicals, is aromatic amines. Example, beta-nephylamines that pass through hydroxylation in the liver to be converted into the conjugated active product. While deconjugation into the inactive component occur in the urinary bladder. So, the aromatic amine is associated with an increased risk of bladder cancer, especially in people working in the rubber and aniline dye industry. Another type of chemicals is nitrosamine. Nitrosamine result from conversion of the deuteronitrates or nitrite into nitrosamine by bacterial gut, so it is associated with an increased risk of gastrointestinal malignancy. The another extrinsic factors are viruses. We have certain type of viruses that are associated with an increased chance of development of cancer, different type of cancer according to the virus itself. So we have hepatitis B and hepatitis C as well are associated with hepatocellular carcinoma, while hepatitis A is self-limiting, does not change into liver cancer. Another type of virus is Epstein-Barr virus associated with Burkitt lymphoma and nasopharyngeal carcinoma. Human papilloma virus, HPV, is associated with an increased risk of development of cervical carcinoma and nasopharyngeal carcinoma as well. Another agent that are associated with an increased risk of malignancy is asbestos. People working in the asbestos industries have a high risk for development of mesothelioma. Aflatoxin associated with an increased chance of developing a hepatocellular carcinoma. Schistosomiasis or bilharziasis may develop a bladder cancer later in life. Helicobacter may be associated with gastric carcinoma and lymphoma, and the other agent is hormones. Higher level of estrogen may lead to development of breast cancer, and similarly androgens may be associated with development of hepatocellular carcinoma. The geographical variation in the incidence of a malignant tumor means that some types of tumor are clustered or aggregated in one part of the world other than the other. Example, so why this is happening? Maybe because of the genetic factor, means local people have a genetic tendency or genetic susceptibility to develop this type of cancer. The other rule is maybe viruses. Viruses are endemic in that part. Example, hepatitis B, Epstein-Barr virus. The parasite or schistosomiasis, bilharziasis is present in this part, especially in Egypt, so it is associated with higher incidence of bladder cancer. The another factor is the diet. Example, in Chaban, we have a higher risk or higher level of development of gastric carcinoma than other part of the world, and this may be related to the diet that people consume in this part of the world. Another factor include hormonal, that is associated with the development of reproductive system tumors. Example, androgen associated with development of liver cancer, estrogen associated with breast cancer. Also, we have a predisposing factors or certain diseases that may be associated or increased chance of developing cancer. For example, ulcerative colitis may increase risk of having colorectal carcinoma, liver cirrhosis, definitely associated with development of hepatocellular carcinoma, and finally, adenoma of the large intestine, which is presented as a polyp. Increased chance of having adenocarcinoma of the intestine later in life. That's why the adenoma is removed. When they found it or discovered by the colonoscope, they removed in order to avoid the risk of malignant transformation. Now we will talk about the molecular basis of cancer. Cancer is a genetic disease. Why? As we said earlier in this lecture, all the discovered cancer have been found to be associated with genetic alteration. Different types of genes are disturbed or altered in these cancer. And you can see all types of dysregulation you can imagine. Deletion, translocation, point mutation, overexpression, underexpression, and silencing. But in order to understand this topic, we will divide the genes into three categories: oncogene, tumor suppressor genes, and DNA repair genes. So these are the genes that are mainly involved in the carcinogenesis. So let's suppose that during cancer we have genomic instability. What does it mean, genomic instability? It means that I have activation of the oncogene and inactivation or suppression of the tumor suppressor gene. These will result in uncontrolled cell growth, and these proliferating cells definitely need a new blood vessel, so I should have angiogenesis in order for the cell to be immotile, to evade all the apoptotic signal, and end up with the invasion with metastasis through blood and lymphatics. And all these are part of the cancer cell hallmark, which is uncontrolled growth, angiogenesis, formation of a new blood vessel, immortality of the cell, lack of response to apoptotic signals, and later cell invasion and metastasis. Now, what do we mean by oncogenes? Oncogenes are normally present in the normal cell, but under the name of proto-oncogenes. They play important role in normal growth and differentiation. While in case of having cancer, they will be changed from proto-oncogene into oncogene through different alteration, either mutation, different kind of mutation, deletion, amplification, translocation, result in formation of the oncogenes. We have different kind of oncogene. The most important examples are C mice, which is normally present as a proto-oncogene, play important role in DNA synthesis. While in case of cancer, the C mice will be amplified or overexpressed, especially in breast carcinoma and neuroblastoma. They have found also that the C mice undergo a translocation in Burkitt's lymphoma between 8 and 14 position. So this is the genetic lesion in Burkitt's lymphoma. Another type of oncogene is RAS oncogene, which is important in the intracellular signaling pathway. So this has been found to be mutated in colonic and lung cancer. Other type of oncogene is HER2, which is a growth factor receptor, is found to be amplified or overexpressed in breast cancer. Now, the tumor suppressor genes. Tumor suppressor genes also present in normal cells, and they play important role in suppression from the name. It's a tumor suppressor gene, so they suppress the growth of the tumor. While in case of cancer, there will be alteration or loss of function of these genes, so they will result in progression of the tumor. The very well-known examples of tumor suppressor genes are retinoblastoma and p53 genes. Retinoblastoma, it's a tumor of the retina. We have two types of retinoblastoma. We have the sporadic and the hereditary or familial. 40% of retinoblastoma are familial. So they occur early in the age, especially less than 1 year of age, and child with retinoblastoma may tend to have osteosarcoma later in life. So, in order to differentiate between familial and sporadic retinoblastoma, as we said, retinoblastoma, in order to have the tumor, the phenotype of retinoblastoma, both copies of the gene should be affected. So, in hereditary retinoblastoma, the baby will born with one copy affected while the other is normal. Then later in life, the patient may have another mutation which result in both of the copies, gene copy affected, and this will result in the development of a tumor. In case of sporadic, the child is born normally, no mutation in either either copy of the gene. Then later, the patient will have a mutation in one copy, and then another mutation of the another copy. This is called the two-hit theory. And as long as the two copies are mutated, this will be enough to produce the retinoblastoma phenotype. The very important tumor suppressor gene is p53 gene. p53 is very important. It is a gene that encodes for a protein that play important role in most, if not all, of the cellular process. It is called the guardian of the genome. It has been found to be mutated or altered or dysregulated in more than 50% of cancer, so it is very important. The location is on the chromosome 17. So why p53 is important? p53 is the center for different types of the outside stresses for the cell. Example, the oxidative stress, nutrient deprivation, hypoxia, DNA damage, any kind of stress. All of these will affect the DNA. The DNA, the p53. The p53 has control over many cellular processes. So, p53 will act through different pathways to govern or direct the cellular processes in a way that is important to keep the cell safe from the external environment. So, for example, according to the external environment, it may activate cellular senescence, angiogenesis, cell cycle, either arrest or activation, migration, apoptosis, or repairing of the DNA damage, whatever, in order to get the cell safe from the external environment and tumor suppression. So get rid or be away from development of the tumor. So, for example, if we have external stimuli, let's say radiation or chemicals, that will result in DNA damage. So, depending on the type of the damage, if it is minor damage to the DNA, then the p53 will arrest the cell cycle in order to give time for the cell to repair itself. Once the damage is fixed and repaired, then the p53 will allow the cell to go through cell division and DNA replication, so you will have a correct DNA that pass from the mother to the daughter cells. However, if we have heavy damage or severe damage to the DNA, that it is irreparable, the damage that cannot be repaired by the cell. So in this case, the p53 will stop the cell cycle and then tell the cell to pass through apoptotic pathway so that the damaged DNA will not pass from the parent cell to the new generation. So in this way, it keeps the fidelity of the DNA replication and get away from tumor progression. In the same way, DNA repair gene act more or less as p53. So they are a group of genes that are important in repairing the damage that may occur to the DNA. Mutation to the DNA repair genes may be associated with a mutation to the oncogene and tumor suppressor gene. So it will result in activation of the oncogene and inactivation of the tumor suppressor gene, which ultimately lead to development of tumor. So, for example, if we have DNA damage, of course, if the DNA repair genes are well and working properly, they will result in stopping the DNA replication and giving time for the cell to repair itself and go through the cell division and DNA replication process. If we have defect in the DNA repair genes, then there will be activation of the oncogene and inactivation of the tumor suppressor gene, which will result in development of cancer through replication of the defective or faulty DNA. يعني الفولتي DNA ده يعبر from the mother cell to the daughter cell. This will result in development of a tumor through the activation of oncogene and tumor suppressor gene. While here, if we have correctly acting genes, DNA repair gene, then there will be stopping of the transcription and replication process, and the cell will pass through apoptosis, so there will be no tumor development. And in the same way, cancer therapy acts by targeting the DNA repair genes. So, mechanism in carcinogenesis. The most important thing in carcinogenesis is that you have to know that the carcinogenesis is a very long-term process. It's not kind of days or weeks. It's very long months, if not years. And we have three processes involved in the carcinogenesis: initiation, promotion, and progression. The initiation starting with the external stimuli that result in genetic modification that affect the genes inside the body. And we know the genes, the oncogene, tumor suppressor gene, and DNA repair gene. Then the promotion, which is the local tissue response to the external stimuli, which depend on the host factor, the age, the immunity, the hormonal state. Finally, the progression of the normal cell into neoplastic cell, and through passing through uncontrolled growth with angiogenesis and passing away from apoptosis, immortality, and later with invasion and metastasis. So the very important thing to know about carcinogenesis is that it's a long process. It's a long time process. And the other thing, it doesn't result from alteration of one gene. It's a kind of several genes affected by the process. The final thing that you have to keep in mind about carcinogenesis, it doesn't depend on the intrinsic factor alone or extrinsic factor alone. It is a kind of interaction between the intrinsic and extrinsic factors that result in development of cancer. So this is the end of the lecture. هي محاضرة شوية طويلة بس مهمة. إن شاء الله إذا رجع الوضع طبيعي وصار عندنا وقت نعيدها راح نعيدها، وأي سؤال تحتاجون أو عندكم سؤال أنا إن شاء الله موجودة. شكرا جزيلا.